So our last speaker comes from Barcelona, Ainhoa Hermoso Mendizabal. Will talk about Striatum, encodes task adaptive expectations, and mediates their impact on perceptual decisions.
So, hi everybody. Today I'm going to present part of my work during my PhD in Jaime de las Rochas lab. So, imagine that we are playing a tennis match, okay? And in the next game, our rival has the net serve, okay? So, we have just a few hundred milliseconds to estimate which is going to be the trajectory of the ball, to prepare the motor plan to return the ball, and to execute it. So, we know that just based on the last series of our rival, that in the 80% of the cases, the rival sends the ball to the left side. So we prepare to return it, okay? But in this trial, our prediction fails and our rival sends the ball to the right corner. Okay, so in these cases that the prediction fails, did we trust all the previous accumulated evidence? Do we continue using this prior knowledge, or alternatively, can we change our strategy and can we dynamically modulate the impact of expectations in our decision? So in today's talk, I want to show you that the rats, they can modulate the impact of this expectation in the decisions, and I will present you the area that is doing this modulation. Okay, so we train rats in the standard reaction and auditory discrimination task, and in order to introduce the impact of prior history, we create serial correlation between trials. In each session, we are going to have two kinds of blocks, the repetitive block and the alternating block. In the repetitive block, the probability to repeat the previous stimulus category is bigger than to alternate, and in alternating block, the probability to alternate is bigger. Here you can see two example sequences. So in order to study the impact of these serial correlations, we are going to analyze the repeating psychometric function. The repeating psychometric function shows the proportion of repeated choices as a function of stimulus evidence in favor of repeating choice. So to this bias, we call the transition bias. Okay, this bias is a history-dependent bias, and it shows the animal's tendency to repeat or to alternate. When it goes to the left, it's positive, and it means that the animals they tend to repeat. And when it goes to the right, it means that the animals tend to alternate. Okay, so what we see that in the repetitive block, after one correct trial, this bias is positive, so it means that the animals in the repetitive block, they tend to repeat, and when they follow correctly several repetitions, this bias builds up until it's saturated in 4 or 5 trials. But surprisingly, after just one error, independently of which was the previous correct sequence, this transition bias goes to 0. Okay, this is what we call the reset effect. Okay, we have this build-up and reset dynamic also in the alternating block with correct alternations and with errors. And this behavior is systematic across all the animals. Okay, so we wanted to find which was the area involved in this build-up and reset dynamics, so we do several bilateral lesions and pharmacological muscimolin activation in different brain areas, and we just found a transition bias decrease in dorsomedial striatum. Okay, so we built a nonlinear generative model in order to see what the animal needs to do to perform the task. Okay, so first, the animals need to capitalize serial correlations, and they need to track all the transitions, and they need to accumulate 10. Okay, for example, in this case, the animal follows 1 repetitions and 2 alternations. So the transition evidence is going to tell the animals that in the next trial should alternate. Okay, but what should alternate? So we need to alternate the previous response that it was right. Okay, so the transition bias is going to tell to the animal to go to the left side. Okay, so imagine that the animal goes to the left side, but it makes an error. So with this model, what we found is that after an error, the transition evidence is not reset, but that it is not projected to the left-right space, that is the transition bias space. Okay, but as the transition bias as the transition evidence is not reset, in the next correct trial, the animals can recover the transition bias. okay? So we can, the build-up effect that I showed you before, the reset effect that I showed you also before, and the rebound effect. This, after just one correct trial, we can recover this bias. Okay, so we want to find these dynamics, okay, in our in dorsomedial striatum. So we fitted this model. I have no time to enter in the details of how we fit this model, but finally we obtain a trial-by-trial estimate of the transition evidence. So I will use these variables, the choice, the transition evidence, and the transition bias, yes, and I will regress them to spike counts using generalized linear models, okay? So I will show you, in the next slides, I will show you neurons that are selective to the choice, to the transition evidence, and to the transition bias, okay? So let's start with the choice selective neurons. So this here is the firing rate versus time, and it's aligned to different epochs of the task. This is a stimulus presentation. This is response onset, when the animal goes from the center port to the left or the right port, and the outcome, okay? So as we can see, this neuron is selective to the left and right choice, okay? And imagine that here resides a reward. So when it's a correct trial, this selectivity, it is maintained until the entire next trial, okay? So, but when the animal makes an error here, this selectivity is not maintained in this neuron. So this is this neuron is representative of what is happening in the population. In the population, what we see is this is the percentage of significant neurons obtained with the GLM analysis, and what we can see is that the percentage of significant neurons increases, and when the outcome is delivered, and just when the trial was correct, it is maintained until the next trial. When it was incorrect, the percentage is not significantly different from the chance. Okay, so in the dorsomedial striatum, just the choice selectivity is just maintained when the previous trial was correct. Okay, so let's see the transition neurons. The transition neurons, as I told you, is the neurons that is selective to the transition evidence. So here you can see the transition evidence that it shows the tendency to repeat or to alternate. So here you can see that this neuron is selected to different transition values, okay, in the next trial before a stimulus presentation. Here I show you the mean rate in 700 in a window of 700 milliseconds before the stimulus, okay? So here you can see the selectivity, and this is when the previous trial was correct. When the previous trial was incorrect, this selectivity is maintained. There is no reset effect, as we expected from the generative model, okay? So this transition evidence is not reset, and is what we see in the neurons. Here you can see another sample neuron, and when we study the population, we see that the percentage of these neurons is around 20%, and that it is maintained during the entire next trial. Okay, so when we see all these neurons, we see that all these neurons are clear and that the information is sustained during the firing rate is sustained during an entire trial. Okay, so the last neurons are the transition bias neurons. The transition bias neurons, this is the same figure as before, this is the mean rate, and this is the transition evidence. Okay, so let's assume that the previous trial was left correct trial. When the previous trial was correct trial, these neurons decrease the firing rate when the transition evidence increases. Okay, but when the previous trial was right correct trial, this neuron increased the firing rate. Okay, with the transition evidence. So if we project this neuron in the left-right space, that is the transition bias space, what we find is that this neuron is encoding the transition bias, the projection of the transition evidence in the left-right space, okay? So these kind of neurons, what we see in the population is that they are maintained until the end of the next trial when it is correct. And when it is incorrect, it is not maintained. Okay, there is no, the transition bias they are not selective to the transition bias. So this is what is causing the reset effect in the behavior that we saw before. Okay, when it is correct trial, we have selectivity to the bias, but when it is incorrect trial, we don't have selectivity to the bias. So let's see this neuron trial by trial, what is happening. So imagine that in the previous trial there was correct trials, so the neuron is selected to the transition bias, but in this trial we do an error. Okay, so the rat does an error, so in the next trial there is not going to be transition bias, so there is not going to be selectivity to this transition bias, but as soon as they do a correct response, this neuron is able to recover the previous transition evidence accumulation. Okay, so these are the build-up, reset, and rebound effect that I explained you in before. So as a summary, we say that we found that in the dorsomedial striatum there are neurons that are encoding the previous correct choices, the transition evidence accumulation, and the transition bias. That is the combination of the transition with the correct responses. Okay, so as summary and as take-home message, when the rat is doing this kind of task, it's accumulating the transition evidence. Okay, the striatum is accumulating the transition evidence. When the rat makes an incorrect response, this transition evidence is updated and is biased, and and generates the transition bias in the behavior. But as soon as they do an error, this transition evidence is not reset, continue maintained, but as previous response is not maintained, the transition bias goes to 0. Okay, but as soon as they do a correct response, as this transition bias, it was not reset, it can recover and it can have like the transition bias. So it does the rebound effect, okay? So I just want to thank you to all the people involved in this project, especially to my supervisor, Jaime de la Rocha, and to Hyafil, who was the college that developed this latent variable model that I used to feed the neural data. So also, I just want to say that I didn't have time to explain how this transition bias and this expectation enter in integration of a stimulus and how are combined with the stimulus. So if you want to see that one, today there is a poster after in the next session, and just say thank you to everybody for listening.
Thanks a lot, Ainhoa, for this interesting talk. It wasn't so clear to me whether this transition bias, whether this is kind of, there's a normative understanding whether this is good or bad, or is it maladaptive, or it was a...
No, yeah. So if you go to here, so this is the necessary bias to do, so the animal accumulates this transition, okay, so we need to accumulate the transition that they have done across several trials in order to increase the performance in the blocks, because at the end we are presenting blocks that is more probable to repeat or to alternate. So in order to do that, to create this bias, they need to project this accumulated evidence of repetitions and alternations. They need to project on left and right space. At the end, they need to repeat or alternate the left and right response. So it's adaptative to the task. This bias is a rule reinforcement bias.
Do we have any other questions? Maybe not. Okay, if not, then thanks for the speakers for the great talks, and thanks to the audience also.